All right. Eve and I are going to try the perhaps ill-advised tag team with two swaps. See how this goes. Um, okay, so the broad question that I'm interested in is that we're interested in is that the natural sounds that we listen to, like animal vocalizations and speech and music, are structured across an enormous range of timescales, and those timescales are hugely variable and stimulus dependent. So our goal is to try to understand how the auditory cortex allows you to flexibly integrate across different timescales. The approach that we're going to take is to measure neural integration windows. So one way of defining the time scale of processing is as the time window that's being analyzed by a neuron or a model, and thus the time window within which stimuli can alter a response and outside of which stimuli have no effect. We've developed a experimental method and computational method to try to estimate this quantity. The basic idea is that we present sets of stimuli. They could be a visual stimulus like a movie or an auditory stimulus in our case. So in this case, they're going to be segments of natural sounds. And we're going to present them in two different orders, such that the same segment occurs in different contexts, which just means that that same sound segment is surrounded by different stuff. So the prediction is that if the integration window is less than the segment duration, then there will be some moment when that window is perfectly contained within the segment, and thus the response, by definition, should be unaffected by the stuff that came around it. In contrast, if the segments are shorter than the integration window, then the surrounding stimuli can alter the response. So I'm first going to show you some modeling results where we basically ran experiments on models. And because we actually haven't had a lot of talks on this kind of thing, but we're going to use deep neural network models because deep neural networks have sort of become the go-to for trying to predict highly nonlinear complex responses in both the auditory and the visual system. I'm going to show you some results that were spearheaded by this very talented PhD student, Manuel Kashishin, where we analyzed units from a deep neural network model, kind of old school at this point, called Deep Speech 2, that's been trained to transcribe speech from sound. And it's just been trained to describe natural speech, which is going to turn out to be important. This model has 7 layers of computation. It's got 2 convolutional layers and then 5 recurrent layers. And from our perspective, the way I think about a layer is just a bunch of model units, and each model unit has a time course. And that time course can be analyzed in a similar way as what you would analyze a real neural response in the brain. I'm going to now describe the key analysis that we're going to use to estimate context invariance. The basic idea is we're going to take the response time course of a model unit or a neuron, and we're going to reorganize that time course as a segment by time matrix. And we're going to do this separately for the 2 different contexts that we test. So the basic idea here is that the first row of this matrix contains the response time course to like the first segment, like the blue segment here, in both of the 2 contexts. The second row contains the response time course to the red segment. The third row contains the response time course to the green segment. Again, this is all just for a single unit. There's nothing multidimensional going on here. We've just taken a single time course and rearranged it as a matrix. Then what we're going to do is we're going to correlate the corresponding columns of this matrix. So at the start of a segment, if we're dealing with a real neural response, which is a causal system, the integration window has to reflect what came before. What came before is the context, and the context is by construction different and independent between the 2 contexts. So we'd expect a correlation of approximately 0. As time progresses, the integration window will start to overlap the central segment. If the integration window is less than the segment, then by definition, at some point, it will be fully contained within the segment, and so the response will be the same, so the correlation will be 1. That makes sense. This is always the hardest. I always should tell people this is the slide you actually have to pay attention to, and the rest will go down like butter. Anyway, okay. I'm going to show you some data from from the DNN. So this is a single example unit from an early layer of the DNN. What I'm showing you is this quantity, the cross-context correlation for different segment durations, 40 milliseconds, 160, 700, et cetera. And the gray region, that's the period where the shared segment was present. So what you see is that the cross-context correlation starts off at 0, and then when the shared segment comes on, it rises and then it falls, as you would predict. And you can see that for segments of about 160 milliseconds, there's a lag where the cross-context correlation gets close to 1, indicating a nearly, though not completely, context invariant response. If you now look at a unit from a late layer of the network, you see a qualitatively similar pattern, but now everything's way longer. So for this unit, you need segment durations of 2 seconds or even longer for the response to become fully invariant to context. So for the model, we can quantify this by just finding the segment duration that's needed to achieve a certain threshold level of invariance. And when we do this, and if we measure this for natural speech, what we see is that this model learns to integrate hierarchically. So the integration windows are short at early layers and long at later layers. And this differs substantially from the randomly initialized network, suggesting that this pattern is learned and not built into the architecture. The next thing that we tested is we wanted to know whether these integration windows actually reflect time, like milliseconds, or instead reflect something about the stimulus, like how fast things are varying, or maybe the duration of the structures that are present in speech, like phonemes or words, etc. So what we did is we took speech and we stretched it or compressed it. So the idea is that when we're doing this, we're changing the rate at which basically everything varies, other than maybe pitch in the stimulus, and we're also changing the duration of all of the structures, all of the phonemes and words, etc. And so if the integration window reflects time, this shouldn't matter. But if the integration window reflects the rate at which information is varying in the stimulus or the duration of important structures like phonemes or words, we should see an apparent change in the integration window that scales with the magnitude of stretching or compression. Okay, so this is one of my favorite plots that we produced in this work. So what I'm showing you here is the integration window for natural speech on the y-axis, stretch speech on the x-axis. The green line is the line of unity, so that's what you'd expect for time-yoked integration. The purple line is the magnitude of stretching on a logarithmic scale. So that's what you would expect if the integration window was changing with the magnitude of stretching or compression. And every dot reflects a single unit colored by the layer. And what you can see is that there's this beautiful transition from short time-yoked integration at these early layers up to long structure-yoked integration at these late layers. And this transition is nearly complete. It goes from nearly fully time-yoked to nearly fully structure-yoked. And we only see this in the trained model. And the model, again, has only ever been trained on natural speech. It has never seen compressed or stretched speech in its life. So it learns this entirely from the variability of natural speech. Okay, so this helps to validate the method, and it provides some predictions for what we might observe in the brain. So let's see what happens in the brain.
Okay, so using artificial networks, Sam has been showing you two key main features of temporal integrations. Hierarchical organizations with longer integrations in deeper layers of the network and yoking to acoustic structures. So our key interest now is to understand whether this is also found in biological networks in the auditory cortex of two species, ferret, I will be talking about that, and humans, so Sam will be coming back on stage for telling you more about that. So a ferret is a good model for multiple reasons, but in this particular study, ferret, so it's a carnivore species, and it has very well described primary and non-primary fields in the auditory cortex, which allows to test the prediction of hierarchical organizations and also to draw some comparison to some extent with the human auditory cortex. So Magdalena, present here, has been a PhD talented student in the lab at the time, has been recording multiple multi-unit and single unit in these two fields of the auditory cortex of the ferret. So this is the top view of the ferret auditory cortex. I'm going to start by showing you raw data. So this is just a scatter plot of one unit recording in primary auditory cortex. And so each row is a repetition of the same stimulus, this orange snippet, presented in one context. And you see reproducible response pattern of that particular unit. Now I'm showing you again a raster plot of the same unit in response to the same sound, the same orange snippet, but in a different context, a different order. So you can see there's a different sounds before. And you see that at first, so just after the sound onset, the onset of the orange snippet, you see the response is different between the two contexts. But after about 50 milliseconds, you see that the response pattern converges between the two contexts, meaning that the units at that time reach context invariance in order that the response of the neuron become invariant to what preceded the orange snippet. This is the same with another unit, and you see that for this particular unit, it takes longer, about 80 milliseconds, to reach context invariance, so after even 100 milliseconds. Okay, so now we're gonna use exactly the same technique that Sam was describing to estimate integration window, with the only difference that in artificial networks, you have no noise. So the noise ceiling is 1. With a neuronal response, obviously, we have a variability across repetitions. So we need to estimate a noise ceiling that will be not 1, but it will be smaller than 1. And we simply do that by computing the correlation across repetition of the same context, the same order. So doing so, this is an example unit from primary auditory cortex. And so you can see in blue the cross-context correlation curve that is increasing throughout the duration of the snippet. So each panel corresponds to snippets of different durations. And the noise ceiling is shown in black. You can see that for this particular unit, the cross-context correlation curve reaches the noise ceiling very fast in about 10 milliseconds. So it's a unit that can reach invariance very shortly, in very short time. So that has a short integration period. This is another unit still from the primary auditory cortex, and you see that for that unit, the blue curve takes longer, about 30 milliseconds, to reach the noise ceiling. So that particular unit takes longer to reach context invariance, so therefore has a longer integration window. Now, if we look at two example units from the non-primary auditory cortex, you see that this unit takes much longer, like 70 milliseconds or so, to reach context invariance. So just from this example unit, it seems that non-primary auditory cortex takes longer to integrate. So to actually extract parameters and measure how long the integration is in these units, we use a model that allows us to extract integration width. So in other words, how long the neurons is integrating over. And so in this eye of bird view of the auditory cortex of the ferret, each unit that we recorded is shown by a dot. And the color scales tell you about the duration of integration. And you can see a gradient from a primary to non-primary regions with a lengthening of the integration window in non-primary auditory cortex compared to primary auditory cortex. That is also summarized here with this histogram of width of integration in primary versus non-primary auditory cortex. So in some, integration windows vary about 4 octaves across the auditory cortex, from 50 milliseconds to 150 milliseconds in the ferrets. And we found, like in the artificial networks, a hierarchical organization across regions. Next, we zoomed in and wanted to know whether also at the level of cortical layers, we would have a hierarchical organizations. So for that, we paired with Stephen David in Oregon that has been performing laminar recordings in the auditory cortex of ferrets also. And the laminar compartment from superficial to deep layers is shown in the color code. And in this histogram, you can see that the localization of the neurons in terms of laminar compartments matters very little for the integration width compared to the regions. So in other words, the main driver of the hierarchical organization of temporal integrations is the region, primary or non-primary, but not much the layers from which the neuron is recorded from. So last, I wanted also to know whether the neurons could adapt to the rate of information of the stimuli. So we performed exactly the same experiment as Sam described. So we presented compressed and stretched stimuli. And in this scatter plot, I'm showing you each dot being a single neuron, the integration width that was measured for the compressed and stretched stimuli. So again, we measure this integration width in the two contexts. And you can see that all neurons are basically on the diagonal lines, meaning that contrary to the artificial network, there is no adaptation to the rate of the incoming information. So this being said, I leave the floor to
Okay, so a human is not a ferret, right? And in fact, Eva and I have a separate project where we've shown that there are quite big differences between the auditory cortex of humans and ferrets due to things like specialization for speech and music. So let's see what happens in a human. So for this, we're going to show you intracranially recorded data from human epilepsy patients. You've already heard a lot about this, so I won't belabor it, and I don't have time to. So here I'm now showing you a map of integration widths estimated from a human. We see kind of qualitatively a similar pattern where there's about three or four octaves of variation, and that there's a clear hierarchical organization where integration windows increase as you go from more primary to more non-primary areas. The main difference is that everything's slower. So the integration widths are maybe twice as long in the human as what we see in the ferret. The second thing that we wanted to test was whether we see a change from time yoke to rate yoke or structure yoke integration, like what we saw in the model, particularly given that there are these regions in the human brain that are very selective for speech, particularly in these non-primary regions. So we ran a similar experiment where we took speech, we stretched it and compressed it. We didn't stretch it and compress it so much that you can't understand it. So all of this speech is clearly intelligible to a human listener. And we did the same thing. And the answer is that we found that, again, basically everything was time yoked. So even in these kind of non-primary regions where you see clear selectivity for speech and where these regions integrate across a few hundred milliseconds, about the duration of a syllable or a word, everything, at least in the auditory cortex, still seems yoked to absolute time. So this suggests that there really are integration windows, right? That's one of the key messages, is that there's this time window outside of which context, including in natural stimuli in an awake listening human, is largely invariant to everything else that came around it. That window really is a property of the neural system, at least in the auditory cortex. It's not a property of the stimulus. And so the kind of direction that we're going now is we're trying to test and trying to understand how the auditory cortex might adapt over longer timescales. Everything we've done so far is, you can kind of think of it as kind of whether the auditory cortex instantaneously adapts to the stimulus. So I have a PhD student, Joseph, who's been trying to run some behavioral and perceptual studies to characterize what we call the podcast effect. So if you ever listened to speech that's been sped up for a while, like for a minute or two, and then you go back to the normal rate, many people anecdotally report that there's a big change in your perception. It starts to sound slowed down. And it's a pretty substantial and notable effect. So we've been trying to come up with a paradigm to quantify this. And I have no time. So I'm just going to, basically, we have 2 minutes. All right, let's do it. We present people with pairs of speech that have it's either at a normal rate or it's been sped up or slowed down. And we ask them to judge which one is normal. And when you do this, you get this nice psychometric curve when you plot performance as a function of the rate of the non-normal stimulus. So if the non-normal stimulus is really fast, they're good. They know it's not normal. If it's really slow, they're good. If it's close to normal, they're bad. Right? So you get this nice kind of U-shaped curve reflecting people's performance. And then what we do is we expose them to an adapter stimulus that in this case is 2 minutes, where they listen to sped up speech for a while. And then we do a single trial afterwards. And when you do this, you see that there's this huge shift in this psychometric curve, which we can fit with a very simple signal detection model that assumes that the rate of speech is changing. And the model suggests that there's about a 20% change. So if you listen to sped up speech for about 2 minutes, it's like your perception of the speech rate changes by about 20%, which is a substantial number. So what we're trying to do now is to kind of characterize this effect behaviorally and perceptually, understand how long it takes to build up and decay, and whether it's speaker specific and all of that good stuff. And then try to understand some of the underlying neural mechanisms that could potentially explain this. So I described the paradigm for measuring temporal integration windows. That's very general. I showed you some evidence of how this works in the network, and then we got into the brain, and I showed evidence that there really is this kind of time-locked integration window that is organized hierarchically across regions, but not layers, and really reflects absolute time and not the stimulus. And finally, I told you about this kind of new perceptual effect that we're looking into. And the stuff I didn't have time to tell you about is Yves is doing all this cool stuff, looking at effects of behavioral and cognitive state and seeing how that affects the integration stuff. So 0 minutes, I'm done.
Okay, we have time for a couple questions.
Yeah, shoot.
Neat thought. I was curious about neural mechanism, and I'll just say that one of the surprises for me was that they were born to wouldn't be different. I was thinking recurrent connectivity or something.
Yeah, right. I mean, one thing that's interesting from the DNN modeling is that, like, if you have just a vanilla LSTM network, you do not see any real change in the hierarchy from a just baseline initialized network. So it's like it's something about the way in which the weights are trained over time is what induces that hierarchy. Yeah, I really don't know how to think about exactly. I feel like this feels like a place where theory is really lagging, is the difference between layer-wise hierarchical computation and region-wise hierarchical computation. And yeah.
This is maybe a similar kind of question, but certainly we know that there are very long time scale dependencies in perception, and I wonder what you think is sort of missing from your model that would allow a system that's only tracking time on these shorter time scales to register those kinds of dependencies.
Yeah, I sympathize with that question. Yeah, I mean,
you want to repeat it?
Yeah, sorry. The question is, like, look, obviously there are longer time scales, so what the heck? Instead of yeah, I think whatever. That's how I take the question. I sympathize with that. Like if you go to the superior temporal sulcus, like the highest stages of the auditory language hierarchy, we have another experiment looking at that, and like you can get up to like 2 seconds, and it's like, well, what about this whole talk that you listened to and integrated at the beginning? I mean, I would say I don't really know. I mean, one thing is empirically, this measure is not really assuming very much, and it's done in naturalistic stimuli. So I think it suggests that these short timescales really matter. I think there's a lot of emphasis on these long timescales, and I'm sure they're important. But at least from the perspective of the neural systems that I've analyzed so far, the data is the data, and it suggests these short timescales are really important. I'd say another one small caveat I'll say is that our noise ceiling requires repetition. Right. So anything that is like totally changing from the first to the second time you hear something, we're not capturing. So that may be part of the story. And thirdly, there may be these long timescale effects, and they may account for a tiny fraction of the variance, right? So anything that explains 2% of the variance is not going to drive these measures, but that 2% could be really important, right? So I think